What are biomaterials and how are they used in medicine? Biomaterials are materials that are used to interact with biological systems, whether in a full organism or in vitro. They are designed to mimic or supplement natural biological materials and functions, and to interact with the body in a way that promotes healing, regeneration, or other desirable outcomes. Applications of biomaterials in medicine have grown rapidly in recent decades, and they've proven to be an essential component in many of the most advanced therapies and medical procedures. Biomaterials can be of various types, ranging from ceramics and metals to polymers and composites, and they are used in a wide range of medical devices and therapies. One of the most prominent and common uses of biomaterials in medicine is in prosthetic devices, such as hip or knee replacements, heart valves, and dental implants, among others. These devices are required to withstand the load and stress of the human body while also remaining biocompatible and resisting corrosion. In prosthetic devices, biomaterials are chosen depending on their properties of strength, ductility, and biocompatibility, among other considerations. For example, the metals used in joint replacement may be titanium or cobalt chromium alloys, while the polymers used in dental implants may be polyethylene or polyether-ketone, PEEK. Another important application of biomaterials in medicine is in tissue engineering and regenerative medicine. Biomaterials can be used to provide scaffolds on which cells can grow and differentiate into complex tissues, such as muscle, cartilage, or bone. This is often necessary in cases where natural healing or repair mechanisms have failed, such as in the case of extensive burns, traumatic injuries, or degenerative diseases. In addition, they must be biodegradable or bioresorbable, so that they do not remain in the body indefinitely, but can be replaced by natural tissues. Examples of biomaterials commonly used in tissue engineering and regenerative medicine include proteins, such as collagen or fibrin, and polysaccharides, such as chitosan or hyaluronic acid. These materials can be processed into porous scaffolds, fibers, or gels, and can be seeded with cells from the patient or from other sources, such as stem cells or donor tissues. With proper design and manufacturing, biomaterial scaffolds can promote tissue regeneration and repair, and even stimulate the formation of new organs. A third area where biomaterials are being used in medicine is in drug delivery. Biomaterials can be used to create carriers or vehicles that deliver therapeutic agents, such as drugs, proteins, or nucleic acids, to specific targets in the body. By controlling the release kinetics and the route of administration, these carriers can improve the efficacy and safety of drugs, reduce the frequency of dosing, and minimize systemic side effects. The choice of biomaterials for drug delivery is based on several factors, including their biocompatibility, their stability and solubility, and their ability to encapsulate or conjugate with drugs. Different types of biomaterials, such as lipids, polymers, and nanoparticles, are being investigated for different applications and targets. For example, liposomes and polymeric nanoparticles are developed for cancer therapy, where they can selectively target tumors and release their payload of drugs in a controlled manner. In addition to the three main areas of application mentioned above, biomaterials also play a role in diagnostic imaging and sensing. One of the challenges in the development and use of biomaterials in medicine is ensuring their safety and biocompatibility. While biomaterials are designed to interact with biological systems, they and also trigger immune responses or toxicity if they are not properly tested or designed. Therefore, biomaterials must undergo rigorous testing and regulatory approval before they can be used in clin. ICAL applications. This includes testing for cytotoxicity, immune response, biodegradation, and other factors that may affect the performance and safety of a material. Another challenge is the cost of biomaterials, especially those that are custom designed or require complex manufacturing processes. This can limit their accessibility and affordability, particularly in developing countries or for patients without adequate insurance coverage. Therefore, there is a need for continued research and development of biomaterials that are both effective and affordable. Advances in biomaterials science and engineering have also led to ethical and social considerations regarding their use. Similarly, the development and use of biomaterials for cognitive enhancement or other non-medical purposes may raise concerns about fairness, safety, and access. In recent years, biomaterials have also been utilized in the field of neurotechnology. Neurotechnology involves the development of devices that interact with the nervous system in order to improve or restore function. This includes devices that stimulate or record neural activity, such as deep brain stimulators, spinal cord stimulators, and neural implants. Biomaterials can play a crucial role in the development of these devices by providing an interface between the device and the nervous system. This interface, known as an electrode, must be able to record or stimulate neural activity while remaining biocompatible and avoiding damage or inflammation to the surrounding tissue. Recent advancements in electrode design have led to the use of biomaterials such as graphene, carbon nanotubes, and conductive polymers. These materials have properties that make them ideal for use as electrodes, including high conductivity, flexibility, and biocompatibility. In addition, their unique physical and chemical properties allow for precise manipulation of neural activity in targeted regions of the brain or spinal cord. The use of biomaterials in neurotechnology has opened up new possibilities for the treatment of neurological disorders, such as Parkinson's disease, epilepsy, and chronic pain. Deep brain stimulation. DBS, for example, involves the use of electrodes implanted in the brain to deliver electrical stimulation to targeted regions. This has been shown to improve tremors, rigidity, and other motor symptoms associated with Parkinson's disease. Despite the promising developments in biomaterials for neurotechnology, there are still challenges to be overcome. One of the main challenges is the development of biocompatible electrodes that can remain stable and active in the body for extended periods of time. The body's immune system can react to foreign materials, leading to inflammation or rejection of the electrode. Therefore, it is important to design materials that are able to avoid or mitigate these immune responses. Another challenge is the development of high-resolution electrodes that can record or stimulate neural activity with greater precision. This requires the use of materials with specific electrical and mechanical properties that can be tailored to the specific needs of different applications. Furthermore, advancements in biomaterials must be made in order to produce more cost-effective materials or those patients who may not be able to afford the most advanced technologies. In summary, the use of biomaterials in neurotechnology holds promise for the treatment of neurological disorders and the development of advanced devices that interact with the nervous system. However, further research is needed to address the challenges associated with the development of biocompatible, stable, and precise electrodes. Advances in biomaterial science and engineering will be key. CAL in overcoming these challenges and unlocking the full potential of neurotechnology. Another area where biomaterials are being used in medicine is in the development of biosensors. Biosensors are devices that can sense and measure biological signals or analytes, such as glucose, cholesterol, or hormones, in a sample of blood or other body fluid. They are widely used in diagnostic testing, disease management, and personal health monitoring. Biomaterials play a critical role in the development of biosensors by providing a platform on which to anchor or immobilize the sensing elements, such as enzymes or antibodies. The biomaterials used in biosensors must be highly biocompatible, stable, and able to promote the adhesion and activity of the sensing elements. They must also be able to withstand the harsh conditions of biological sample and remain responsive to the analyte of interest. They offer several advantages over conventional diagnostic tests, including real-time and continuous monitoring, portability, and minimal sample requirements. Challenges in the development of biomaterial-based biosensors include the optimization of the sensing elements and the biomaterial platform to achieve high sensitivity, specificity, and selectivity. Thanks for watching.